Hello everyone, my name is Geeta here, going to present the topic genotoxicity as per the OECD guideline under the guidance of C.R. Patil sir. So, the content of my topic is starting with the history and background and ending with the references. History and background for the genotoxicity study. So, there were the scientist Gregor Mendel, who is known as the father of genetics. He conducted the experiment in pea plant in 1956 and he found that plant receives the genetic information from its parent which passed down throughout the generation. And in 1927, there were the scientist Muller. He discovered the role of radiation in producing the heritable changes in living organisms. Then after that, there were the scientist Orbach who first discovered the ability of chemical to cause the mutation. Then, by considering the genotoxicity study, the OECD has developed some test guidelines for studying the genotoxicity in 1987. So, here the word OECD stands for Organization for Economic Cooperation and Development. Here, the different guidelines used in the genetic toxicity study are OECD guideline, as I already mentioned about it, then ICH guideline and Schedule Y under the D and C Act, India. So the introduction for the genotoxicity. Genotoxicity, the word itself indicates that the genotoxicity here the gene means any DNA material or the genetic material and toxicity means any damage to that DNA material. So this genotoxicity word encompasses the term mutagenicity and DNA damage, which result in permanent alteration in the structure or information content of the surviving cell or its progeny. So there are some chemical substances which is known as the genotoxins, which is also known as the mutagen that cause the genotoxicity and leading to the damage to the DNA or chromosomal material and which leads to the mutation. Then OECD has developed the guideline for assessing the genotoxicity in chemicals. Then genotoxicity involves various in vitro and in vivo tests designed to identify any substance which may induce the damage to the genetic material. There are some agents that cause the direct or indirect DNA damage, like reactive oxygen species, UV and ionizing radiation, then nucleoside analog, topoisomerase inhibitor, and protein synthesis inhibitor. These all reagents leads to the direct or indirect damage to the DNA material. So this DNA damage leads to the activation of the feedback mechanism of our body. So there are some feedback mechanism like checkpoint activation, DNA repair mechanism. There is a mismatch repair, nucleoside excision repair, base excision repair, homologous recombination, and non-homologous enjoining and direct reversal and apoptosis. So the activation of all these feedback mechanism, these systems recognize the whatever the DNA damage occurs and that damaged DNA will be corrected through these feedback mechanisms. Here, the DNA damage leads to the genotoxic risk. If that DNA damage occurs into the somatic cell, it leads to the some pathological condition like aging, cardiovascular diseases, and cancer. But if that genetic toxicity or DNA damage occur into the germ cell, it leads to the sterility problem into the male and female. There are also the chances of some genetic diseases like single cell mutation. It leads to the cystic fibrosis, sickle cell anemia, and hemophilia. Then, after that, there are the chances of the multifactorial diseases like psychosis, diabetes, and cardiovascular diseases. The purpose of a OECD guideline for the genotoxicity study is to provide a framework for evaluating the potential mutagenic and genotoxic effect of the chemical on living organism. This guideline covers various aspects of the genotoxicity testing. Here, the different types of tests are conducted and whatever the evaluation criteria required for those tests are also taken into consideration. Then also the specific guideline for nanomaterials are also provided. Then potential use of new approach methodologies in genotoxicity assessment are also taken into consideration. So there are some mechanism of the genotoxicity. So there are the chances of the direct DNA damage, indirect DNA damage, and epigenetic modification. So if there is the direct DNA damage occurs, so there are the some mechanism like single and double strand break, then loss of excision repair, cross-linking, alkali lapile site, point mutation, and structural and numerical chromosomal aberration. And in the second, indirect DNA damage. This mechanism involves the production of the reactive oxygen species, and that reactive oxygen species or the molecule leads to the DNA damage. This is the indirect mechanism. And third one is the epigenetic modification. This epigenetic modification involves the DNA methylation and histone modification. But specifically in epigenetic modification, there is no structural change into the DNA. Means no any single base pair is replaced and altered. But there is the methylation and histone modification. This mechanism leads to the DNA damage. Then this is the pictorial presentation of the genotoxicity mechanism that I already mentioned. There are some test guideline that according to the OECD guideline, which is starting with the test guideline 471, which is known as the bacterial reverse mutation test, which is also known as AMIS test, and it was adapted first into the 1983 and currently it is adapted into the 2020. Then the guideline that I have mentioned into the yellow color, these are already deleted. But whatever the guidelines are remaining, those are already in use. So let's start with the in vitro test for genetic toxicity. So first test is the AIMS test, which is also known as the bacterial reverse mutation test. So the history behind this test is that in 1970s there were the scientist Bruce Ames, who is the American biochemist and geneticist. He developed the Ames test to identify the substance that caused the mutation. Introduction. Ames test is widely used method developed by the Bruce Ames to evaluate the mutagenic potential of a chemical compound. This test specifically involves the use of the some strains of a bacteria like of the Salmonella typhimurium and Escherichia coli bacteria to test whether the chemical is having the mutagenic potential or not. This AIMS test involves some procedure like first the selection of a test organism, then exposure of that test organism to the test substance, then replica plating is there, and at the end result interpretation is there. Principle of the test method. Suspension of the bacterial cell Salmonella typhimurium is exposed to the test substance in the presence and absence of the exogenous metabolic activation system. Here is the metabolic activation system will activates the metabolic processes of that cell. In this method, there are the two methods, plate incorporation method and plate incubation method. In the plate incorporation method, this suspension are mixed with an overlay agar and then immediately plated onto the minimal medium. But in case of the pre-incubation method, the treatment mixture is first incubated and then it is mixed with the overlay agar before plating onto the minimal medium. So after the plating, it is incubated for 3 to 2 days, 2 to 3 days. Then after the 2 to 3 days, the revertant colonies generated are counted and it is compared with the spontaneous revertant colonies generated into the solvent control plate. So if here the revertant colonies generated are more into the positive control plate as compared to the solvent control plate, it indicates that the chemical is having the mutagenic potential and it is not suitable for the use. The description of the method. First, we have to consider the preparation. So, there is the specific requirement that at least we have to select the five strains of a bacteria for this test. So, there are some examples of the strains like Salmonella typhimurium test strain 1535, then Salmonella typhimurium test strain 1537, test strain 97, 97A, 98 and 100 are selected for the test. Then there are some strains of the E. coli bacteria. Example WP2 UVRA or E. coli WP2 UVRA PKM 101, and then Salmonella typhimurium test strain 102 is also used, but it is rarely used. The next is the medium for the Ames test is the Vogel-Warner minimal medium E and glucose. Then overlay agar containing histidine, biotin or tryptophan. Here the metabolic activation system is it is specifically derived from the mammalian origin. Typically, a rat liver S9 hepatic fraction is selected as a metabolic activation system. This system stimulates the metabolic processes. This system activates the pro-mutagen. So here we know if the pro-mutagen is get activated, it will be get converted into the mutagen, and if it is converted into the mutagen, it leads to the genotoxicity, and it also making it more sensitive and relevant to the potential mutagenicity of a chemical compound. So next is the test substance preparation. So there is the requirement that the concentration of a plate should be 5 milligram per plate or the 5 microliter per plate of a test substance. Then next one, there are some control test substances are selected as a baseline comparison. Example 910 dimethyl anthracene, 712 dimethyl anthracene, congorate, benzopyrene and cyclophosphamide. So next one is the procedure for pre-incubation method. Pre-incubation method involves the pre-incubation of test mixture. Then after the pre-incubation in that test mixture, 0.05 to 0.1 ml of bacterial suspension will be added. Then in addition, there is the requirement of the metabolic activation system. So the volume required is the 0.5 ml, and after the all the addition, there is the need of incubation. So that incubation generally needs for the 20 minutes at the 30 to 37 degrees Celsius. After the incubation, it is added into the overlay agar and pour into the surface of the minimal agar plate. After that, it is also incubated and the number of revertant colonies generated per plate in negative and positive control medium are compared, and at the end, standard deviation should be calculated. Here also the same principle that if the reverted colony generated into the plate are into the positive control medium are more as compared to the negative control medium, so it indicates that the chemical is having the mutagenic potential, and if the vice versa case is observed, then we can indicate that the chemical is not having the mutagenic potential. So this is the pictorial presentation of the pre-incubation method. Here we have selected the test strain of Salmonella typhimurium, which is histidine negative strain. Then it will be added into the plate. In addition, as I already mentioned, that there is the need of the metabolic activation system, which will be extracted from this rat liver extract. Then after that, it will be added into the plate. Then incubation will be done. After the incubation, again the number of revertant colonies generated into the positive control medium and negative control medium will be evaluated. Next is the plate incorporation method. First, we have to prepare the culture of Salmonella histidine auxotrope. Then mix that auxotrope with the test substance in molten agar. Then pour that molten mixture on the top of agar plate and incubate at 37 degrees Celsius for 3 to 4 days. Here, until the histidine level depleted all the histidine cell will grow in the presence of rest mutagen. But as soon as the histidine level is completely exhausted from the medium, only the revertant colonies will grow. At the end, if higher the number of colonies, greater is the mutagenicity. Pictorial presentation of the plate incorporation method. As here the Salmonella oxidrops are selected, then those are mixed with the overlay agar. Then after that it is incubated for a particular period of time. Here we incubate it for the 2 to 3 days, and after the 2 to 3 days, the number of revertant colonies generated, those are compared with a negative control plate. So as I already said, if the revertant colonies generated into the positive control medium are more, and as compared with the negative control, so that indicates that the R chemical compound is having the mutagenic potential. And if it is the vice versa case is observed, then it indicates that the chemical is doesn't having the mutagenic potential. Next is the data and reporting. So, as we follow the GLP, there is the need to mention whatever the step you follow, then whatever the procedure you use for the test. So, all that data generated or the whatever the results are generated, that you have to make the report of that procedures. So similarly, in the genotoxicity study, there is need to mention whatever the data generated during the study. So here at the end, there is need to report the whatever the individual plate count observed into the test, then mean number of revertant colonies per plate, then standard deviation, dose response relationship, and statistical analysis. So all that data generated should be reported. So here is the formula for calculating the mutagenic ratio. So mutagenicity ratio is calculated by this formula. So uh, MR means the mutagenicity ratio is equals to the spontaneous revertant colony generated into the negative control plus induced revertant colony generated into the positive control medium, then again divided by the spontaneous revertant colonies. So this formula will give the information about the mutagenicity ratio. So if that mutagenicity ratio is more, means it indicates that the chemical is showing the mutagenic potential, and if it is showing the zero value or the very less value, it indicates that the chemical is doesn't having the mutagenic potential. So next is the in vitro mammalian chromosomal aberration test. So here the this original test guideline 473 was adapted in 1983. This test is used to identify the substance that cause the structural chromosomal aberration into the cultured mammalian cell. So this structural aberration is of two type, chromosome type and chromatid type. So here the chromosome is a DNA molecule with a part or all of the genetic material of an organism. Then chromosome are the vector of heredity. But chromatid is a one copy of a newly copied chromosome which is still joined to the original chromosome by a single centromere. Next is the principle behind the chromosomal aberration test. Here we have to select the cell culture of human or other mammalian origin, and then it is exposed to the test substance or the test chemical with and without the exogenous source of metabolic activation system. After that, it is incubated for the particular period of time. After that, it is treated with a metaphase arresting substance. Example is the colchicine and colchicine. Then, after the incubation, it is harvested and stained with a particular intercalating dye, and metaphase cells are analyzed microscopically for the presence of chromatid type and chromosome type of aberration. Next is the description of a method. So there are the different cell lines that we have to use for this test. Example is the Chinese hamster ovary, then Chinese hamster lung, then human or other mammalian peripheral blood lymphocyte. Next is the media and culture condition. So, media that we are going to use for the test, it should be appropriate, and the culture condition, it should be also appropriate. Here, the culture condition should be maintained at a 37 degrees Celsius. Next is the preparation of a culture. As we are using the different cell lines, I already mentioned about it. Um, Next is the lymphocyte. If we are using the lymphocyte for this test, it is needed to treat it with the heparin to avoid further inconvenience during the test. Next is the metabolic activation system, which is needed for the cell growth. After that, the test chemical preparation. So here, if that test chemical is present into the solid state, we have to dissolve that particular chemical into the particular solvent according to the need. And then if that test chemical is available into the liquid form, we can use it as it is. Also there, if there is the need of dilution, then also the selection of a appropriate solvent should be there. The solvent that we are using for this test or the dilution purpose, it shouldn't show any reaction with the chemicals, or it should not give any unwanted result or the unwanted chemical reaction with the other ingredient into the medium. Next is the control test substances. Example is the clastrogen active without the metabolic activation. Example, methyl methane sulfonate, mitomycin, 4-nitroquinoline, N oxide, cytosine arabinoside. Then clastrogen requiring metabolic activation is the benzopyrene and cyclophosphamide. Procedure for the chromosomal aberration test. So here the treatment of a test substance with lymphocyte started at 48 hours after the mutagenic stimulation. Then it is exposed to the test substance with and without metabolic activation for 3 to 6 hours. After that, it is sampled at an equivalent about 1.5 normal cell cycle length after the beginning of a treatment. Then after that, chromosomal preparation step is there. Here the culture is treated with the colchicine or the colchicine for 3 hours prior to the harvesting. Also the hypotonic treatment of the cells, fixation and staining is there. After that, analysis is done by the microscopic analysis, and during this we have to record the polyploidy and endoreduplication. At the end, treatment result should be mentioned as the percent of cell with a structural chromosomal aberration. Data and reporting. Here, a percentage of cell with a structural chromosomal aberration should be evaluated. Then different type of structural chromosomal aberration should be listed with their number and frequencies for experimental and control culture. Then percentage of polyploidy and endoreduplication cells are reported. And at the end, evaluation and interpretation of a result. So whatever the results are generated, it should be evaluated for the chromosomal aberration test. So this is the mammalian bone marrow chromosomal aberration test, which comes under the test guideline 475. Introduction for this test is: this original test guideline was adopted in 1984 for the detection of chromosomal aberration induced by the test compound only in the bone marrow cell. Here, the animal is first exposed to the test substance, then it is treated with a metaphase arresting substance. Example is the colchicine and colchimide. An animal is sacrificed at the end after the particular time. After the sacrification, bone marrow cells are isolated and examined for the chromosomal aberration test. Here, if the chromosomal aberration will observed in the bone marrow cell, it will indicate that the R chemical is showing the mutagenicity or the genotoxicity. So this is the detailed procedure for the chromosomal aberration into the bone marrow cells. We have to select at least the 5 animal, and it is treated with a test substance. After that, here we select the dose as low, medium, and high. So after this treatment, after the 24 to 48 hours, animals are sacrificed by CO2 euthanasia. Then this bone marrow, sorry, the bone is isolated, means femur is isolated and flush the bone marrow with isotonic solution. After this, the suspension is centrifuged and it is prepared. Then the smear is prepared, and after this, it is treated with some intercalating dyes like GIMSA or the fluorescent DNA specific dyes. Example is the acridine orange, hochist, and pyronine white. So after this staining, it is evaluated microscopically for the presence of the chromosomal aberration. This is the pictorial presentation of a chromosomal aberration into the bone marrow cell. First, we have to select the animal. We have to select the rat or the mouse. Then it is treated with the test substance. After the test substance treatment, femur is isolated. Then it is flushed with the FBS. So FBS stands for the fetal bovine serum. Um, then bone marrow suspension is prepared. It is again centrifuged, and after the centrifugation, smear is prepared on the slide, and then fixation is done by using the methanol. After the fixation, it is treated with the some intercalating dyes like GIMSA staining is there. Then after the staining, it is evaluated microscopically for the presence of the chromosomal aberration. So at the end, analysis would be done. Mitotic index should be determined as a measure of cytotoxicity in at least 1000 cells per minute. In vitro mammalian cell micronucleus test. So, introduction for this MNT is used to determine if the compound is genotoxic to the cell in culture by evaluating the presence of micronuclei. So, the test can be performed in primary human peripheral blood lymphocyte or established cell line such as Chinese hamster ovary or the TK6 cells. So here is the picture of the micronuclei formation. So this small part or the small dot is indicating that the cell is formed with the micronuclei. Principle: cell cultures are exposed to the test substance with and without metabolic activation system. Then it is treated with a cytochalasin B for blocking the cytokinesis. Then cell cultures are allowed to grow for sufficient time. It will allow the chromosomal damage and lead to the formation of micronuclei. Then after that, cells are harvested and stained and analyzed microscopically for the presence of micronuclei and scored. Most commonly used cell lines for this test are the Chinese hamster lung, then Chinese hamster ovary, then Syrian hamster erythrocyte, and V79 and 51781. So these all cell lines are used for the micronucleus assay. So here, this is the procedure for micronucleus test, which is the in vitro test. Here, we have to select the cell line plus culture media for it. Then those cells are taken from the stock solution to prepare the suspension. Here, the monolayer of a cell will be formed. Uh, if we are selecting the blood sample, first we have to treat it with the lithium heparin to avoid the further inconvenience in the test, means to avoid the blood clotting. After that, the separation of lymphocyte will be done and then phytohemagglutinin will be added, which will induce the cell division. Cell culture is added to the cell line with a test chemical. The 3 to 6 hours control medium is with the without S9 fraction and test medium will be with the S9 fraction plus aerochlor 1254 will be added, which will induce the metabolic enzyme into the medium. Then both the control and test medium will be exposed to the test for the 3 to 6 hours, and then a medium will be removed after the washing by the fresh media. Then cytochalasin B will be added. Then after that, the harvesting is done for 1.5 to 2 cycle length, and that cell will be examined at the end for the presence of micronucleus. If the cell is showing that the presence of micronucleus is there, means the it is indicating that the chemical is having the potential to the genotoxicity or the mutagenicity. And if the micronucleus formation will be not there, then it will indicate that the chemical is doesn't showing any mutagenic potential and it is safer for the use. Here, this is the procedure for micronucleus test. Uh, Initially, we have to select the lymphocyte culture, then treat it with the test substance, and then cytochalasin B will be added. After the treatment with the cytochalasin B, cells are harvested and evaluated or examined for the presence of micronucleus under the microscope. So the next is the in vivo genetic toxicity. Mammalian erythrocyte micronucleus test. So here we will select the mammalian erythrocytes for the micronucleus test. So the this test comes under the guideline of 474, which was adapted in 1983. So this is used for the detection of damage induced by the test chemical to the chromosome. The test evaluate the micronucleus formation in erythrocyte. So the purpose of test is to identify the substance is causing the cytogenic damage which result in the formation of micronuclei. Principle behind this MNT test is the test involves the formation of micronucleus. If we are selecting the bone marrow, is then animal is first euthanized, and then after the appropriate time, bone marrow is extracted and preparations is made. But when we are using the peripheral blood, then blood is collected at an appropriate time after the treatment and preparation are made, and it is stained. So after the staining, again the same procedure we have to evaluate for the presence of micronuclei. So the detection is done by microscope, flow cytometry, and laser scanning cytometry. So this is the pictorial presentation of MNT. Here, if we use the clastrogen, it is the substance which induces the structural chromosomal aberration through the breaks of DNA. So these are the different cell cycles. Means cell is undergoing the different cycles of the cell division or the cell formation. After this, during the cell cycle or the cell division, there is the formation of a micronuclei. If that chemical is showing the potential of formation of a micronuclei, that means the chemical is having the genotoxic potential. And here these are the immature erythrocytes, and some are the mature erythrocyte. So here the polychromatic erythrocyte is known as the immature erythrocyte, and the normochromatic erythrocyte is known as the mature erythrocytes. So means specifically if the micronuclei formation observed, means chemical is showing the genotoxic potential. Mechanism of micronuclei formation. So there are the different mechanism. Like there is the mitotic loss of the acentric chromosome, then fragment formation forms the structural aberration, then mechanical consequence of the chromosomal breakage and exchange, such as lagging chromosome, inactive centromere, or the tangle chromosome. Then again the mitotic loss of whole chromosome, then apoptosis. Micronuclei is induced by the clastrogen and anagen. So as I told about, the clastrogen is the substance which induces the structural chromosomal aberration, and anagen is the substance which induces the numerical chromosomal aberration through interaction with the cellular target other than the DNA. such as protein involved in segregation of a chromosome during the mitosis or meiosis. Next is the description of a method. Here, the preparation, we have to select the animal species, we have to maintain the animal housing and feeding condition because there are some epigenetic factors which can affect the results of test. Then preparation of a test animal for the test, then preparation of a doses, then metabolic activation system selection is there, then test condition we have to maintain. Here the solvent and vehicle selection should be appropriate. It should not show any reaction with the other chemical ingredient which are used for the test. Then positive and negative control. So example is the mutomycin and cytosine arabinoside. Next is the mammalian alkaline comet assay. So this comet assay is under the guideline of the 489 of the OECD. So introduction for this, the Swedish researcher Osling and Johnson developed this technique in 1984. So this comet assay is also known as the single cell gel electrophoresis. It is the method used to measure the DNA strand damage and repair in single individual cell when it is exposed to the test substance. Principle behind this comet assay is that the best loops of DNA break and lose their supercoiling, and it becomes free to extend toward the anode when it is exposed to the current during the electrophoresis at high pH. The result appear as a structure resembling comets observed by the fluorescence microscopy. So comet consists of different heads and tails. So the head portion is composed of the intact DNA, while the tail is contains the damaged DNA or the broken pieces of the DNA. So the intensity of a comet tail is relative to the head reflect the number of DNA breaks. Means if the tail portion is showing the more number of the damaged broken pieces of the DNA, that means there is the formation of the comet, and that comet indicate the then length of the comet indicate that the chemical is showing the genotoxic property. Procedure for the comet assay. First, we have to prepare the slide. So, agar is mixed with the low melting agarose at 37 degrees Celsius. Then, cell suspension will be prepared. It is immobilization of a cell on the slide will be there. Then, cell lysis. Cell lysis will be done after the treatment of that cell with the particular detergent or the chemical. So here the histone and here the DNA will be separated from the histone and it will be free for the electrophoresis process. During the electrophoresis, unwinding will be occur when it is treated with alkali. So DNA unwinding and denaturation will occur. After that, the staining and comet scoring will be there. So staining will be done by using the some intercalating dyes and visualized by the fluorescence microscopy following the alkaline electrophoresis. So this microscope will be get connected to the particular software by using the high resolution camera, and the comet form is observed under the microscope. So if the head portion is showing sorry, the tail portion is more, means it indicates that the uh, number of damaged DNA or the broken pieces of DNA are more as compared to the head portion. So there are the different software which are used for the Comet Assay, like Comet Analyzer, Comet Assay IV by Instant, and AutoComet. So by using this software, we can evaluate the comet form during the study. So here we have to just transfer the live video picture using a high resolution video camera, which will be attached to the microscope. Then here we can select 50 randomly overlapping cells per sample. Then it will be scored. When using the comet assay IV, we have to click on each cell in turn and key comet assay parameter, which will be including percent tail moment, percent length, and percent hedgehogs. It will be recorded. So here the actual procedure that we have to follow for the comet assay. So after the treatment with the chemical, oxidative stress will be generated and it leads to the DNA damage. So whatever the DNA damage occur, it will be evaluated by using the software like the AutoComet software or the uh, Comet IV software. So here is the microscope. This microscope will be attached to the high resolution camera, and then through this camera it will be attached to the software. The picture which will be generated under the microscope, it is evaluated for the comet form. So here the intensity of a head portion and tail portion will be evaluated. If the tail portion is showing the high length, means percent tail length is more as compared to the head part, it indicates that the that tail portion is having the more number of the broken pieces of DNA, which at the end indicates that chemical is showing the genotoxic property or it is causing the genetic mutation. So here the structure of comet is there. This portion is this intact portion which can be observed here. That intact portion is indicating that the intact head is there. Sorry, intact DNA is there, and this blurred part is showing the tail portion of a comet, which will show the intensity of a broken DNA pieces. So the test include two distinct in vitro method like thymidine kinase gene in L5178Y mouse lymphoma cell. Then next one is the hypoxanthine guanine phosphoribosyl transferase gene in Chinese hamster ovary cells. For this, that cells means the cell line will be exposed to the test substance, means chemical treatment is there. Then if there is the mutated HPRT gene is there, then there is the loss of enzyme activity and that cell will follow the de novo synthesis pathway, and at the end cell get mutated and it gets survive into the medium. But if there is the no mutation occur after the treatment with chemical, so there is the active HPRT enzyme is available and it will follow the salvage pathway, and there is the integration of a test gene into the DNA and ultimately it leads to the cell death. So it indicates that the test substance is having the genotoxicity property and it leads to the cell death. That means the our chemical substance is not suitable for the use into the body. So here, genetic toxicology, rodent dominant lethal test, which comes under the guideline of 478. Introduction for this. The original test guideline 478 was adapted in 1984. The purpose of lethal test is to investigate whether the chemical can produce the mutation due to the chromosomal aberration in the germ cell or not. Dominant lethal test can cause the embryonic or the fetal death, means dominant lethal mutation cause the embryonic or fetal death A limitation of this test is that it requires large number of animals, it is more laborious, expensive and time consuming process. This test is most often conducted in mice and rats. Principle for this test is that genetic change can occur in the form of structural and numerical chromosomal aberration. Here, in this test, female and male are treated with the test agent. Then male are mated with a group of untreated female. Females are killed after the 14 days after mating and dissected, and after that the score for corpora lutea, then earlier fetal death and total implantations. Procedure for this, first the individual males are mated sequentially at an appropriate predetermined interval to one or two virgin female. The number of matings following the treatment should be governed by the treatment schedule. Then ensure that the germ cells are mutated. Then females should be sacrificed in second half of progeny and uterine content examined to determine the number of implant, live and dead embryos. The ovaries may be examined to determine the number of corpora lutea. Then evaluation is done by the statistical method. This is the pictorial presentation of overall in vivo and in vitro genotoxicity testing for the AIDS test, HPRT assay, then comet assay, chromosomal aberration assay, micronucleus assay. So these are the pictures of whatever the results can be obtained at the end of the test. Application of a genotoxicity study. It provides the information about safety and genotoxicity of newly developed pharmaceutical and chemical. Also, it is used to study the protective effect of the phytochemical on gene. Also used in the area of cancer research for the evaluation of a chemotherapy, and also used in monitor the environmental contamination of a genotoxin, and assessment of oxidative stress with various human diseases, nutritional studies measuring the DNA repairs. So these are the references for the genotoxicity study. So there are the different test guidelines are available on the OECD website that we can follow for the detail.